Hello, everyone. I'm Jeremy Wilkinson, PacBio segment lead for Microbial Genomics, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the recent advances by PacBio and Zymo Research in microbiome and metagenomics that are leading to improved results, capacity, and cost effectiveness in microbial community profiling and mag assembly. So I'm joined today by two great researchers who will present in the following order after my presentation. Dr. Annabelle Dameron, a scientist at Zymo Research, and Dr. Daniel Portick, a staff bioinformatics scientist at PacBio. So we have a lot of great material to cover today, and the presentations portion of this webinar is pre-recorded, but will be followed by a live question and answer session with all of the speakers. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat or at the end. We've also uploaded a number of complementary pieces of literature to your control panel, so please feel free to download them and take them with you. We will be recording this webinar and making it available for download in the next few days, so please keep an eye out for a follow-up email with a link to the recording. Immediately following this webinar, you will receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. Also, for those that attend this webinar in its entirety and fill out the survey, we will select a winner for free PacBio HiFi sequencing project to be run at Zymo Research. You can also stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. So with that, let's get started. All right, so first I'd like to give a brief overview of PacBio sequencing and how it elevates microbial genomics. So as I assume you may know, PacBio is providing solutions for complete and accurate long-read sequencing, most recently with our Sequel IIe system. And while we've done so for well over 10 years now, we've seen particular enthusiasm and adoption through the development of our HiFi reads. These HiFi reads are sequence reads that are uniquely both long and highly accurate, so 99.9% and greater. And as you'll see today, the Q scores for metagenomics are often in the Q40s, and for amplicons and 16S, they are often around Q60. And these have really been demonstrated to have a powerful utility in many areas of genomics, whether it be genome assembly, detecting all variant types, targeted sequencing, long-read RNA sequencing, or what we call isoseq, or metagenomics. So HiFi reads really provide a new lens to interrogate living systems and thereby allow for many discoveries that are simply not possible with the other technologies. And in today's presentation, we'll highlight examples using HiFi sequencing for microbiome and metagenomics research. Okay, so HiFi reads have taken the best of both worlds that previously existed. That is short, accurate reads from Illumina and long but error-prone reads like ONT and PacBio was previously. HiFi reads combine both the long read lengths. An average HiFi read is about 100 times longer than your typical Illumina short read, and a high sequence accuracy into one single technology. So HiFi reads are typically 15 to 20 kb in length, and the raw polymerase reads in PacBio sequencing have become so long now, certainly well over 100 kb. This allows for building consensus from multiple passes on a SmartBelt insert template, which leads to the 99.9% and greater in accuracy of the HiFi reads. And the sequencing errors here is indicated as these red dots. They are random, and therefore they'll be washed out by increasing the number of passes around the SmartBelt template. And there are many publicly available HiFi metagenomic datasets available to you on our GitHub that you can check out here. The mean read lengths range from a variety of different lengths from 5 kb up to almost 20 kb, and that really depends on the insert size that goes into the library prep. And again, the median quality scores that you see here range from Q30 to above Q40, which represents up to 99.99% accuracy and higher. So there are really four primary ways that you can use PacBio HiFi sequencing to understand your microbial community and microbial species. These are full-length 16S sequencing or full-length ITS for fungi or other types of targeted approaches. And this really reveals the true diversity of your samples with better taxonomic resolution at the species and strain level versus what you can get with short read alternatives, and generates more testable hypotheses. And then with HiFi shotgun metagenome profiling, it provides more and richer functional information without the need for error correction or assembly, and really reveals the true positives in taxonomic profiles with high precision. And then for metagenome assembly, HiFi reads provide far more high quality metagenome assembled genomes or MAGs than other technologies. and more complete circular and single contact mags, even at lower coverages. And an important point is that short read polishing or hybrid assembly is no longer necessary with HiFi. And there are many studies that have come out to showcase that. And for microbial whole genome sequencing, so from single isolates, you can get complete microbial genomes with ease and confidence. And for most microbes, closed genomes with accessory plasmids can be assembled with a single touch using the default settings of our assembly pipeline. And again, short read polishing or hybrid assembly is no longer needed when you use HiFi. And all of this is made possible because PacBio HiFi combines both long read lengths with high accuracy. So 2022 is really a breakout year for HiFi in regards to metagenomics, and this spans across a variety of shotgun metagenomics, whether it be assembling mags or for taxonomic or functional profiling. And this is across a variety of different sample types as well. And so just in 2022, there were 15 plus highlight publications showcasing this application for HiFi, and many more publications that focused on amplicon sequencing, such as full-length 16S or full-length fungal ITS. And on the 16S front, we recently put out a press release announcing that we are going to expand our MOS-seq technology, which is our technology that concatenates multiple shorter fragments, such as the 1.5 kb of 16S, into one read on the system, so it acts as one long HiFi read. So this is going to increase the throughput of 16S sequencing on HiFi by anywhere as much as 10 to 12-fold increase on the throughput for the number of reads as well as the number of samples. And also last year, Nature Methods classified long read sequencing as the method of the year, and this is really backed by the work that went into the telomere to telomere project and the publication that came from that, where long read sequencing was really the key player driving that research and actually finally getting the first complete human genome. And so I talked earlier about the Sequel IIe platform, which is our current platform available, but we recently just launched the Revio system, which is our new HiFi platform that actually just had the first instrument placed just a few weeks ago. And this has really given us HiFi sequencing at scale. So what I mean by that is it's now able to do 100 million ZMWs per run, compared to previously with the Sequel IIe, 8 million. And the run times are 24 hours versus the previous 30-hour run times. And the HiFi yield per run is 360 gigabases compared to previously with the Sequel IIe, it's somewhere around 30 gigabases. So this really was designed from the ground up to deliver HiFi at scale. And so when talking about scale, particularly for the ZMWs, these are 25 million ZMW smart cells, and there are 4 of these that run simultaneously on the machine in independent stages. And so that's where the 100 million ZMW output comes from. And again, it's a 24-hour cycle time that all of this takes place in for the 4 independent smart cells. As far as ease of use goes, it's a 50% reduction in the consumables that go into the actual sequencing, has load and advanced capabilities, and importantly, there's no longer a nitrogen requirement. For compute power, we have increased the relative compute power by 20 times. This is now a GPU-based architecture, and it now has Google Deep Consensus on board to assist with the computation. As far as affordability goes, it's only 330 USD per 1 gigabase HiFi genome at 30x coverage. And it's less than a minute to load the instrument. And also because there's this increase in data, we've now done a greater than 50% decrease in the actual file sizes that are created off the platform because of new compression capabilities. So a little bit more about the 4 independent stages. They really enable flexible experimental designs. These can be different sample types or different library types. So here's just an example scenario for this area of research. So you could have full-length 16S on cell 1. On cell 2, you could have shotgun metagenomes. On cell 3, you could have fungal ITS. And on cell 4, you could have microbial WGS for single isolates. And just an idea of what you can expect as far as throughput goes for metagenomes, for mag assembly in particular. So mag assembly requires a lot higher depth than shotgun profiling. So on a per year basis, the Revio platform, because of all these things I just talked about, it's able to produce 4,000 metagenomes per year for mag assembly in order to recover high quality mags for around the top 100 species or strains in the metagenome sample. Whereas the Sequel IIe, for that same amount of high quality mags per sample, it would only be 250 of those. So that's a 15 times increase in the throughput that the Revio is able to do compared to the Sequel IIe. Plus, this throughput is even more so for metagenomics for the purpose of taxonomic and functional profiling and not assembly, which for profiling purposes, again, it requires less number of reads. So that would actually be another 25x of that 4,000, which would be around 100,000 metagenomes per year for profiling on the Revio system. And lastly, I wanted to mention our Discoveries Roadshow, which is happening right now. So we are coming to multiple cities in North and South America and in Europe, as well as APAC, which is going to be announced very soon. So we are bringing you scientific talks, network opportunities, different updates on our technology. You can scan this QR code now, or you can visit pacb.com/events and find Discoveries Roadshow and register there. And with that, thank you for your time.
Hi, my name's Annabelle Dameram, and I'm a scientist at Zymo Research focused on metagenomics data analysis. Today, I'll be discussing how to optimize your sample preparation workflow for long-read metagenomic sequencing. Just a quick rundown of what we're going to cover in the next 10 minutes. So we'll start with an overview of the metagenomics workflow, specifically focusing on sample preparation. We'll then touch on some of the challenges and solutions here before we review an optimized workflow for long-read metagenomics. Traditionally, in microbiology, we focus on characterizing one or a few organisms at a time, whereas with metagenomics, we're interested in the entire community present. So there are different challenges we must consider. The metagenomics workflow can be broken down into two parts: sample preparation involving study design, which includes the use of appropriate standards, sample collection and DNA isolation, and sequencing, which begins with library preparation, followed by downstream bioinformatic analyses. And I'm going to be discussing sample preparation today. At this crucial step, there are some important factors we must consider. When collecting a sample, we need to be confident that the composition of the sample is not changing between the point of sampling and DNA isolation, as this could impact the overall outcome of our study. We also need to preserve DNA integrity to ensure sufficient quality for sequencing. When isolating DNA, we must ensure uniform lysis of all microbes present to ensure we're obtaining an unbiased profile. And for long-read sequencing applications, DNA length is another key consideration. As I touched on in the last slide, sample collection and preservation are crucial but are often overlooked. Without sufficient preservation, the taxonomic profile of a sample can change over time due to microbial growth and decay and nucleic acid degradation. If we take this burger, without preservation, the profile would look very different compared between day 1 and 1 month later. A common method to preserve samples is to freeze in liquid nitrogen and store at -80. However, this method is not always convenient if samples are being collected out in the field in remote locations, or for example, if a patient is self-collecting a fecal sample and there will be a delay in getting this sample to the lab. An alternative is to collect your sample into a preservative reagent, such as DNA RNA shield, which can inactivate microbial growth in the sample and enable storage at ambient temperature. And we offer various products for convenient collection of a variety of different sample types. These graphs each show the profile of the same fecal sample over a 1-month period, with the coloured stacked bars representing the relative abundance of different bacterial phyla, without a preservative reagent on the left or with on the right. And we can see that without preservation, the abundance of phyla changes over time, and we see a bloom in the pink bar corresponding to proteobacteria, whilst with preservation, the profile remains unchanged. And this is important because if we were to process our sample even 1 week after storage, we would draw very different conclusions about the organisms present and their relative abundance than if the sample was processed on day 0. The benefit of the Shield preservative reagent is that it also preserves DNA integrity during the DNA isolation step. This tape station image shows that we obtain higher molecular weight DNA with less fragmentation when samples are pre-treated with Shield preservation reagent versus a typical PBS buffer. When isolating DNA from a metagenomic sample, an analogy we like to use is that each organism is like a different nut, which we must crack open to reveal the genomic information inside. And certain nuts are harder or more difficult to crack open or lyse than others. For example, mammalian cells which do not have a cell wall are easier to lyse compared to bacterial cells which are encased by a cell wall. Focusing on bacterial cells, gram-positive bacterial cell walls depicted on the right have a considerably higher mechanical strength than the cell walls of gram-negative bacteria on the left, due to the presence of a multi-layer peptidoglycan cell wall depicted by the purple circles, and this likely makes them more difficult to lyse. Fungal cell walls not shown here have an even more complex cell wall structure and are also generally accepted to be more difficult to lyse. This means that it's important that the lysis method we choose is able to consistently crack open both those easier and harder to lyse cells. The most common lysis methods include the use of chemicals to permeate the cells, using enzymes to digest the cells, or the use of agitators to mechanically force open the cells to release the nucleic acids inside. However, both chemical and enzymatic lysis methods are selective in that they are best utilized for pure cultures and so must be validated for each species. However, mechanical lysis is the least biased approach, enabling indiscriminate lysis of cells, and so it's best suited to metagenomic applications. We found that optimal lysis efficiency could be achieved following 40 minutes of lysis using a vortex genie, and we selected the vortex genie as a commonly accessible lysis tool. Importantly, we found that DNA fragment size following mechanical lysis can vary based on the lysis machine used. For example, higher energy homogenizers like the FastPrep, shown by the orange arrow, will result in reduced DNA fragment size compared to lower energy homogenizers like the Vortex. Of course, when we're considering long-read metagenomic sequencing, DNA length is critical. And when using mechanical lysis protocols, there has traditionally been a trade-off between efficient lysis and DNA length, which is a major challenge for long-read sequencing. We have both manual and automated kits for DNA isolation, which utilize unbiased mechanical lysis and produce DNA fragments which are of optimal length for HiFi sequencing within the 8 kb to 20 kb range. Up until this point, we've discussed optimizing our sample collection and preservation and our DNA isolation methods. But a key consideration is, how do we know if our protocols are optimal and unbiased? And this is where standards and controls come in. Standards provide a basis for comparative evaluation and allow us to establish trust in the quality of our analysis. Take this snowboarder. Without any point of reference, it's really impossible to determine the height of their jump. But using standards and controls, we can not only evaluate the performance of our protocols, but we can also ensure consistency between different experiments in our own lab and even compare experiments run by different labs, facilitating multi-lab collaboration. One example of a standard is our whole cell mock microbial community. This is a reduced complexity microbial sample, which is comprised of both a mix of those harder to lyse organisms, like the gram-positive bacteria, and easier to lyse gram-negatives. And the key point is that each organism is present at a known quantity. So we can include this standard in our workflow to test if our abundances of these organisms match the expected. And we can see here that using certain protocols, gram-positive bacteria can be insufficiently lysed, meaning that they are underrepresented in our results. So looking more closely at the composition of the mock microbial community, there are 5 gram-positive, 3 gram-negative, and 2 yeast species. We have a range of standards which each fit a different purpose. So besides from the whole cell mock community discussed on the last slide, which can be used to detect bias in lysis and extraction. We also have a pure DNA extract of this standard, which can be used to evaluate library preparation. We also have a true diversity reference, which is in the form of a real-life fecal sample, which you'll hear more about from Dan in the next talk. And this can be used to evaluate downstream bioinformatic analyses along with detecting biases in the whole workflow. And Dan is going to present some fantastic results in characterizing the metagenome of this sample. We also have various spiking controls, which can be especially useful to test the limits of detection of organisms and the sensitivity of your workflow. To conclude here, an optimized workflow for long-read sample prep should consider sufficient sample preparation, which can be achieved using a preservative reagent, unbiased and efficient lysis, which yields DNA of the appropriate length for HiFi long-read sequencing, and should consider microbiome standards to enable benchmarking and consistency across experiments. Thank you all for your attention.
Great. Well, I'm excited to be here today. My name is Dan Portick. I'm a staff bioinformatics scientist on the computational biology group here at PacBio. And today I'm going to tell you about advances in HiFi metagenomics, and specifically how we're using these to characterize a really interesting pooled human gut microbiome reference. So in the webinar today, I first want to cover this new reference material, how it was produced and what we want to use it for. And then I'm going to focus on two main topics, and those include read-based taxonomic and functional profiling and metagenome assembly. And for each of these topics, I'll give sort of a high-level overview, cover some new methods developments and workflow developments, and then show some results from the analysis of this sample. So in metagenomics, we often use mock communities, and mock communities are artificially created data sets where we know the species and relative abundance design. And so mock communities are really useful for benchmarking to assess consistency across technologies and labs. They're also really useful for providing a ground truth for methods development and testing. But the reality is that mock communities are often far too simplistic in terms of the species and relative abundance designs. And so they have limited utility in terms of these uses for benchmarking, methods development, and other applications. And so this gap has led to the development of a new reference material. This is the Zymo fecal reference with true matrix technology. And so this was produced with a collaboration between BioCollective and Zymo. It's a pooled cohort of human gut microbiomes. And so it's more diverse than a singular human gut microbiome in terms of species diversity. It's also going to have a lot more strain level variation too, which is important. And so it is a truly complex alternative to mock communities. And the great thing is there's over 2 million aliquots available. And because of the way it was produced with the true matrix technology, the material is consistent across these different aliquots, making it broadly available and useful for the research community. And at PacBio, I am really interested in understanding what's in this community. And so as an empirical standard, this is a little bit different because we don't have a ground truth for which species are present and their relative abundances. But with deep sequencing, we can sort of attempt to fully characterize the sample. And I'm going to be showing how we're doing this using taxonomic profiling as well as metagenome assembly approaches. So let's talk about some of the data sets we've generated for this sample. And so we have two distinct HiFi data sets. The first was a deep sequencing experiment that we ran on our Sequel IIe system. It involves sequencing 6 8M SMRT cells, which yielded about 121 gigabases of total data. This comes from about 17 million HiFi reads with an average length of about 7 kb and a median QV score of 40. And that represents 99.99% accuracy. But recently in January, we were able to sequence the sample on our new sequencing platform. This was Revio. And so we sequenced two 25M SMRT cells, which yielded 135 gigabases of total data, about 17.6 million HiFi reads, similar read lengths, and actually a higher QV score than what we attained on Sequel IIe. So in addition to these two HiFi data sets, one of the things that I'm kind of interested in is what are the effects of total data on downstream analyses? And so to sort of get at this question, I artificially downsampled the Sequel II data set. So from 6 SMRT cells, I sort of bioinformatically downsampled this to represent 5 SMRT cells, 4, 3, 2, 1, all the way down to a 48-plex. And so I'll be talking about not only these full data sets, but also this sort of downsampling data set throughout the rest of the talk. So let's now move into our first main topic, and this is read-based profiling and classification. So here the goal is to identify the species present in a sample and estimate their relative abundances. Now in the figure on the right, let's assume we have a microbiome. We've done bulk DNA extraction. We've then done shotgun sequencing. Let's assume these are HiFi reads. We can then take those HiFi reads and align or match them to some type of a reference database. This can be nucleotide sequences, proteins, or k-mers. Once we have all those matches, we can then interpret them and assign them to taxonomy. And ideally, what we want at the end is a taxonomic profile where we have several species or genus level categories, and then we have sort of read counts for each of these individual taxon categories. Now, there are several challenges associated with profiling and classification. One of the most important ones is that a lot of the methods that were developed specifically for long reads were developed very recently. And so the best practices and sort of how these methods perform was unclear because comprehensive benchmarking had not yet been performed. And there's also always this difficult balance between precision and recall. So how can we minimize false positives while also being able to detect species at very low abundances? And so to address these kinds of questions, I recently performed a study which was published at the end of last year in December, and we really tried to perform a comprehensive benchmarking study of classification and profiling methods specifically for long-read shotgun metagenomic data sets. And we found some really interesting results. And we were able to identify some methods that performed really well. So the figure on the right is showing the relationship between precision and recall. And the top right corner of that figure is high precision and high recall. And that's where the ideal methods are. And we can see that there are several that occur in that zone. So in terms of the best trade-off for precision and recall, we found sour mash was the top performing method. In terms of perfect or near perfect precision with slightly reduced recall, this involved Diamond and Megan LR, as well as BugSeq. So these were the three methods that we identified as top performing and we would highly recommend. Now, there are some differences between these methods. So for example, sour mash is k-mer based, whereas Diamond and Megan LR is based on protein alignments, and BugSeq is based on nucleotide alignments. One thing I want to point out about the Diamond and Megan workflow is that because we're aligning to proteins, we can generate a really nice taxonomic profile. But if these proteins have known functions, we're also simultaneously assigning functional annotations. So we can build a functional profile as well as a taxonomic profile. And that's currently the only method that's capable of doing that simultaneously. In terms of workflows, we developed PacBio pipelines for sour mash and Diamond and Megan, whereas BugSeq is actually a PacBio compatible partner. So they have a cloud-based platform where you can do online submissions and download reports. So you can choose which method works best for your use case depending on sort of the sort of desired outcomes as well as computational resources. But all three of these methods work extremely well for PacBio HiFi data. So let's jump into some of the results. So here I'm showing the results for those three methods. What I'm showing here are the numbers of species detected at each of these different abundance categories. And in the top row, we have ultra low abundance, and towards the bottom, we have higher abundance, so greater than 1% relative abundance. And what I want you to focus on first are these methods in gray. So these are sort of the optimized filtered methods, which are really tuned for high precision. And we can see that we detect anywhere from 155 to 223 species based on these filtering criteria. And in white, I'm showing some unfiltered results from these different methods. And what you can see is those numbers jump up quite a bit. So up to greater than 7,000 species with no filtering. But you'll notice most of those assignments are happening in these ultra low categories. And here we can have a partial match to a single read, which is counted as a species. And based on our study, we've quickly identified that a lot of that is noise and there's very little signal there. So this sort of demonstrates the importance of using these threshold or internal filters in these programs to obtain high quality, confident results. Okay, so these are just some general numbers. What do these profiles actually look like across the methods? So rather than showing you a heat map of over 300 species, I tried to simplify it by moving up a taxonomic rank to the genus level. So here what I'm showing are 77 genera, which were detected at greater than 0.05 relative abundance by one or more of the methods. Dark colors represent higher abundance and lighter colors represent lower abundance. And if you look to the left of the figure, you can see that there is some pretty good concordance across the methods in terms of relative abundance. We see bacteria that we would expect in high abundance, like Bacteroides, Faecalibacterium, Glaudia, and some of the other common players. But you'll notice there are also some differences between the methods. One of the things that's not totally clear, as these analyses are pretty recent, is whether these are actual differences detected between the methods, or if this is more of an artifact of differences in taxonomy. And so when we use different taxonomies, we can get these incompatibilities that have nothing to do with biology, but just are related to nomenclature. And so this is something that I'm working on cleaning up at the moment. We'll sort of figure out how consistent these profiles actually are. So what are the effects of total data? So for this, I use the artificially downsampled data set, and I use the Diamond and Megan pipeline to investigate this. So I have 4 cells of data down to a 48 plex. So this represents 90 gigabases down to 0.5 gigabases. What was really interesting to see is that not only did I detect the same number of species, but also really consistent relative abundances too. And so we can get the same information from 60,000 HiFi reads, 0.5 gigabases, as we can with 4 cells of sequencing. And so to me, what this implies is that because these long reads are packed with many genes, when we use methods that take advantage of that information, we don't need a ton of reads to build a highly accurate profile. And so this really opens up the door for using shallow shotgun sequencing to generate highly accurate taxonomic profiles. And this is something that we're really excited about. And the last thing I really want to touch on in this section is related to functional profiling. So again, as I mentioned, the Diamond and Megan workflow will output functional annotations in addition to these taxonomic annotations. And what's really incredible is that because HiFi reads are packed with gene content, approximately 92% of the reads received at least one functional annotation. But in reality, it was actually more of an average of 2 to 4 annotations per HiFi read. And that's not just 2 to 4 genes on a HiFi read, but 2 to 4 genes with a known functional annotation per read. So there are many additional genes present there that we just simply can't match. And what's really cool is that over 67 million functional annotations were assigned across all the 4 databases that Megan interrogates. And so again, this shows really the kinds of information that we can gather from individual reads. When we aggregate that across large data sets, we can build really diverse, highly accurate taxonomic as well as functional profiles. Okay, so let's move on to the second main topic of today, which is metagenome assembly. So here, the goal is to assemble a representative genome for each microorganism present in the original microbiome sample. And so we tend to call these representative genomes MAGs, or metagenome assembled genomes. And this distinguishes this type of sequencing and assembly from something else like, let's say, isolate sequencing. There are a lot of challenges associated with metagenome assembly. So first of all, there are multiple species present. That's not true for most targeted sequencing projects. Second, these species are present in uneven and unknown coverages. And finally, there are repeat regions and intergenomic overlaps that occur across species, as well as between strains of the same species. And so I would actually suggest that metagenome assembly is still by far the most difficult assembly task out there. But the good news is long reads can really help with this problem. And so there are important differences between short read versus long read metagenome assembly. And I want to kind of highlight what that looks like here. So in this figure, I'm showing what a typical short read assembly project would look like. So we do our sequencing, we end up with millions of reads that range from 50 bases to 150 bases. When we perform our assembly with these short reads, we end up with tens of thousands of contigs. And these tend to be on the small to perhaps medium size range. It is exceptionally rare, if not impossible, to assemble a complete circular contig for any of the species present in the original sample, just due to the challenges that I listed in the previous slide. And so as a result, we have lots of fragmented genomes in this contig set. We have to rely on binning methods to identify and group these contigs that belong to the same putative organism. And any time that we group lots of contigs together, we have opportunities to make mistakes. And so at the end of this process, we end up with what I would describe as draft quality mags, where each mag is composed of anywhere from 50 to 500 contigs. And although the single copy genes might be there in the levels that we expect, we might be missing large parts of the accessory genome that we're completely unaware of. So these are highly fragmented, potentially incomplete draft quality mags, which is less than ideal for what we want to use in comparative studies. And when we use HiFi metagenomics, we're starting at a different point. So the thing that I really want to emphasize here is that the HiFi reads themselves are often the same size or larger than the contigs that we obtain through short read assemblies. Right, so we're starting this assembly process at a fundamentally different point. And what we find is that we routinely get circular contigs straight from the assembly, which represent complete bacteria or archaeal genomes. But for the same reasons, low abundance species or challenging genomes, we will get fragmented mags as well. The good news here is that the contigs tend to be much larger, so hundreds of kb to megabases in size versus really, really small. And so this makes binning a lot easier. But long read assemblies also will benefit from the binning process. We use that to extract these additional fragmented mags. And in the end, we end up with what I would describe as very high quality mags. So some of these are single contig with high depth of coverage, and others are composed of a handful of contigs. And in the beginning of 2022, we actually published a new algorithm for metagenome assembly that is specifically tuned to HiFi reads. And this is called HiFi ASM Meta. And in the publication, we demonstrated that HiFi ASM Meta produces more total mags, and more importantly, more circular mags than any of the other methods that are currently available. And so this represents a tremendous improvement and a leap forward in terms of the metagenome assembly process. But you don't have to take my word for it. Let's just look at one of the assembly graphs. So this is for the pooled gut microbiome sample. This is the Zymo reference. And here, if you're looking at the assembly graph, what you might notice is there's lots of circles. And in fact, if you look closely, there are over 56 circular contigs that I have highlighted here. And these are all greater than 1.5 megabases. And so these are likely to represent complete bacterial or archaeal genomes that come straight from the assembler. But you'll also see that there are plenty of linear contigs here. And this again is the reason why we would want to use binning approaches so that we can identify and extract these additional high quality mags from this contig set. And so to do that, we need tools for post-processing. And over the last year and a half or so, I've been continuously working on the HiFi Mag Pipeline. And so this is a post-processing pipeline that takes assembled contigs and outputs the high quality mags. I just released a new version about 2 weeks ago. It implements what I would describe as a completeness-aware strategy. And this is highly effective at obtaining mags, and I kind of want to run through what this looks like. So we know that we can get complete bacterial and archaeal genomes straight from the assembler. It's really important to identify these first and remove them and protect them so that we don't end up misbinning them and tossing them out by accident. So the first step takes all contigs greater than 500,000 bases and individually assesses them for completeness using checkm2. Anything that's greater than 93% complete, we move it to a dereplicated bin set where each of these complete contigs is in an individual bin. Anything less than 93%, we sort of discard. We move that back to the pile of incomplete contigs. And so there, we're taking these incomplete longer contigs and then those shorter contigs to create one FASTA file of what is incomplete contigs. We then take that incomplete contig set and then we perform binning only on those contigs. And here we're using MetaBat2 and semi-bin2 using long read settings. We compare and contrast, dereplicate and merge those using DAS tool. And so then we move those bins into the dereplicated bin set as well. We run checkm2 on everything. We screen out things that are not complete enough or have too much contamination. And for all those bins that pass, we consider these mags. We assign them to a taxonomy using the Genome Taxonomy Database Toolkit. And the final product is high quality mags, sequences, metadata, and visualizations. And the last thing I want to point out about this pipeline is that it represents a tremendous improvement over single binning strategies such as MetaBat2 by itself, and even previous versions of this pipeline that were focused more on things like circularity. And so you can see that the gains here are quite a large percent increase, not only in the total mags, but also these single contig, highly complete mags. And so this is really sort of the state of the art, leading edge in terms of binning algorithms that's highly useful for long read assemblies. So I took our two HiFi datasets, the Sequel2 and the Revio datasets for our sample, and I ran these through HiFi ASM Meta and the HiFi Mag Pipeline. And I recovered 186 or 189 high quality mags. And below, I'm showing you some of the characteristics of what those mags look like. So the figure on the left is showing the relationship between completeness and contamination. Each dot represents an individual mag, and they're color coded based on the number of contigs that they contain. And ideally, what we want to see is a bunch of yellow dots in the bottom right-hand corner of that figure. And this is a zone of really high completeness and really low contamination. And if we look, we actually find that there are 67 mags here with what I would consider superior quality. Single contig, greater than 90% complete, less than 5% contamination, which is pretty incredible. And if we look at the figure on the right, this is showing the relationship of the genome sizes of those mags versus the average depth of coverage. And so we have genomes that span from 1 megabase to 6 megabases with an average depth of coverage anywhere from 5x to almost 800x. And these mags are actually color-coded by their GC content. And so we have mags that span from 30% GC content up to 70% GC content. So lots of different variation in terms of genome size, in terms of GC content, and we're able to assemble these mags with pretty high success rate. So of those 189 individual mags, I classified them into about 154 species. And so one of the interesting things is that 14 of these mags cannot be classified to the species level, and in some cases to the genus or even family level. And these are likely to represent novel diversity in the human gut microbiota. And this is something that I'm particularly excited about. But in addition, there were 19 species that are represented by 2 to 3 mags. And so what this implies is that there's strain level variation. And I'm showing you an example of this down below. So I obtained 2 mags, which represent Akkermansia muciniphila. When I align these to the genome taxonomy database reference and made dot plots, you can see that there's some structural variation as well as SNP variation across the length of the genome. And that's even true if we compare the mags to each other. Even though they are highly similar, there's some differences there. And so the really good news is that when we use highly accurate long reads and methods like HiFi ASM Meta, we can actually assemble strain level variation into the respective genomes that they belong to. And this is really an important leap forward as well in terms of metagenome assembly. So everything that I just showed you was using binning based on bioinformatics algorithms. So we're using things like tetranucleotide frequency and average depth of coverage, or even some machine learning models to group these contigs. But those are all just sort of approximations. There are other strategies now that offer improved ways to bin. One that I am very excited about is offered through Phase Genomics, and this is their Proxy Meta platform. So here, we're using proximity ligation within individual cells to sort of create these Hi-C libraries. And so what's happening internally is that we're sort of attaching the bacterial chromosome to itself in different ways to get proximity information. But in addition, anything else that's in the cell is also going to be sort of grouped in there too. So this includes things like mobile elements, so plasmids and virus. And when you prepare this library and you sequence it, you then gain access to all this proximity information. And you can actually use it to improve binning because we know which pieces of DNA are actually touching each other. And you can also associate these mobile elements to their hosts. And so we work with Phase to generate library and sequencing for the Zymo sample. And the results were really incredible. So if we look at the Sequel2e and the Revio dataset separately, with Sequel2e, I was able to obtain with best-in-class binning, the HiFi Mag Pipeline, 186 total mags. With Proxy Meta, that jumped to 269. And with Revio, I was able to obtain 190. With Proxy Meta, that number was 313. So we're seeing a 44 to 64% increase in total mags, which bioinformatically, we're never going to catch up to. But in addition to just increasing numbers, we're also increasing quality. So again, these binning algorithms are sort of approximations and they can make mistakes. But with proximity information, we know which pieces of DNA are touching each other. So these bins are much higher quality and much more trustworthy than some of these other algorithms, even though we've come a long way in terms of binning algorithms in general. But in addition, we're also getting these other types of information. So things like virus host associations, as well as AMR to host associations, so antimicrobial resistance genes. And so we get this really rich complementary data set in addition to improving our binning. And so I'm really excited to kind of dive into this in a little bit more detail and see how we can more fully characterize the sample in terms of virus and plasmid associations too. So one of the common questions we get is how much sequencing do we need in order to achieve good assembly results? And so this is kind of a moving question. So it kind of depends on the diversity of your sample, but it also can be approximated. And so to do this, I looked at the downsample data set. What I did is I took each of these different data levels, ran them through HiFi ASM Meta and the HiFi Mag Pipeline. And here I'm showing you the total number of mags that came out the other end. And what we see is actually a fairly predictable relationship between sequencing depth and total mag yield. And so even at an 8-plex, we're obtaining 17 mags, which are likely to represent the things that are present in the most high abundance in the sample. That number quickly climbs when we go to 1 SMRT Cell at 111, and it sort of plateaus a little bit as we increase sequencing depth because we're sort of accumulating those really low abundance taxa, and we're able to assemble those genomes. So at some point, we would expect this to level off. Now, high sequencing depth isn't necessarily ideal if you want to do lots of samples, right? But the good news is Revio is completely changing how we're thinking about this. So what used to be a deep sequencing project is now becoming easy and accessible and routine using Revio. And so the dot on the very far right represents what came out of the Revio dataset, which was just 2 of those 25M SMRT Cells. So higher data yield and more mags than we can achieve with 6 8M SMRT Cells on Sequel IIe, which is right next to it. And if we look kind of towards the center of the figure, what we see is Revio with 1 SMRT Cell is the equivalent of 3 SMRT Cell 8M on Sequel IIe. And there we're obtaining about 174 mags. And just sort of as an interesting exercise, I took the number of mags, divided it by the cost of consumables for sequencing with the different platforms to come up with a price per mag. And here with Sequel IIe, it's about $22 per mag for that 174 mags. But with Revio, because we're increasing throughput while dropping costs, it actually turns out to be about $6 per high quality mag. And so we can achieve much greater throughput. We can do multiplexing. We can do deep sequencing for relatively low cost. And so I think with Revio, we're going to see a lot of projects unlocking. We're going to see more adoption of HiFi sequencing for this purpose. And that's something that I'm very excited about in the next year. So to summarize, today we talked about a few different topics which are related to characterizing the species composition of this new pooled gut microbiome sample offered by Zymo. And so with regards to taxonomic and functional profiling, I found anywhere from 155 to 223 species in high precision mode. But without any filtering, we can get, of course, crazy numbers over 7,000 species. Lots of functional annotations if you're interested in that information. And most importantly, consistent taxonomic profiles across data levels. From 0.5 gigabases to 100 gigabases, we get the same consistent profile. And with regards to metagenome assembly, I can recover up to 189 high-quality mags using the state-of-the-art PacBio HiFi Mag Pipeline. But with Phase and Proxy Meta, we can recover much more information. And so this yielded up to 313 high-quality mags. And the last thing I want to emphasize again is that deep sequencing for metagenomics is now cost-effective with Revio. You can run more samples with higher throughput for less cost. Finally, all of the pipelines, workflows that I've discussed are freely available on GitHub. We have a Pacific Biosciences GitHub site and the PV Metagenomics tools repos where these things live. There's plenty of documentation and additional resources and links to data sets so you can try out these analyses for yourself. So I would encourage you to go and visit and check out what we have to offer. And finally, if you're interested in long-read metagenomics, please reach out to us directly. My information is below. I'm engaged in a lot of collaborative research projects. I'm interested in developing new tools and addressing really difficult problems when it comes to metagenomics. So please feel free to reach out. And with that, I want to thank you for your time today. And I think we are ready to move on to our Q&A session.
We have a lot of questions. We've answered several in the chat, but we will continue to answer some more here in these last 10 minutes. So here's one that just came in that we can answer. So this one's for Annabelle. How to increase DNA yield when isolating low biomass samples from things like swabs?
Yeah, this is a good question and something that comes up a lot. And what we like to tend to do is work directly with you and your sample type to really make sure that we're getting the best possible yields from your sample. So yeah, this would really depend on what kind of yields we're talking about, how low, we're talking, but definitely it would probably be most helpful for you to reach out with us directly. We could kind of work with you with your specific sample type and give you some customized tips there.
Awesome, thank you. And then this last question that just came in, I can answer. So it's have you an experience with amplicon metabarcoding, i.e. ITS regions for fungi? Yes. So there's lots of publications now where folks have used full-length fungal ITS amplicons sequencing with PacBio, as well as 18S, 28S, basically any gene. You can do the entire operon if you want to. You can do 18S, ITS, and then 28S too, with primers that set across the entire operon. Same thing for bacteria. You can do 16S, ITS, and 23S. Another question for Annabelle. So can you use DNA and RNA field on whole specimens such as mosquitoes?
Yes, so the answer would be yes. You can you could preserve your sample, your whole sample, but you would still later then need to use a lysis method to prior to extraction.
Thank you. Dan, what kind of sequencing depth would you recommend to aim for long-read metagenomic sequencing?
Yeah, that's a great question, and it sort of depends on your analysis goals. For taxonomic profiling, you could do up to a 48-plex on one 8M SMRT Cell on Sequel IIe. That would give you roughly 60,000 reads, about 0.5 gigabases per sample. And we found that that's enough for highly accurate profiling. For assembly, of course, though, we would want a higher sequencing depth. And so the general recommendation is to start out with one sample on one SMRT Cell, so you can sort of get a sense for your diversity and relative abundances within that sample. And you can sort of adjust from there and either multiplex or perform deeper sequencing if you need to get to those ultra low species.
Great, thank you. Here's another earlier one for Annabelle. So does Zymo Research have DNA extraction kits for feces or any clinical material which is not based on magnetic beads?
Yeah, so you have a range of different kits that you could opt for if you didn't want to use magnetic beads. So we have a spin column kit, and we also have a spin plate kit. So yeah, if you were more interested in using a column-based methodology, we also have kits for those purposes.
Okay, thank you. Here's one that I can answer. So what is the average cost of 16S sequencing using HiFi? Depends on the number of samples you're utilizing, but right now the cost of materials and everything, if you have the machine, is somewhere around $10 USD. That's the cost for the amplification, library prep, and sequencing. And that's getting around 10,000 reads per sample. With the MOS approach, like I mentioned in my intro, we're going to be driving that further down at about half that cost. So I'm just trying to filter through those last questions. Okay, so here's one. The Zymo Research metagenomic sequencing services offer DNA extraction. We would like to send raw samples in Shield.
Yes, absolutely. You can send us your samples in Shield and we'll happily perform the whole rest of the workflow through for you all the way through to bioinformatic analyses.
Awesome, thank you. Here's one I can answer. Any comments on HiFi sequencing and viral metagenomics? That's a great question and is of more interest nowadays, obviously. There have been now, I think, at least 5 papers that have come out in the last year that has specifically done this. So they're able to identify and assemble also viral genomes from metagenome samples using HiFi. You can reach out. I'm happy to share those publications with you afterwards as well. Annabelle, how to use DNA RNA Shield for urine samples?
Yeah, so you could use a 2x concentrated Shield, or we also have a UCB buffer that you could use specifically for urine preservation.
Thank you. Dan, what packages would you recommend for mag assembly and binning?
Yeah, that's a great question. So the field is moving quickly right now. HiFi ASM Meta is the best choice right now for HiFi metagenomics. MetaFly is also a really good alternative to HiFi ASM Meta. For binning, I would highly recommend using our strategy where we're focused on completeness aware binning. If you use a simple binning strategy, oftentimes complete contigs can get misbinned and tossed out in the analysis. That's something to be aware of. And I've also been testing semi-bin too with the long read settings, and that's been working extremely well for our use cases. So I would recommend using the PacBio specific tools. Those work for long read assemblies across the board.
Great, thank you. Here's one I can answer. So what's the estimated cost for shotgun metagenomics? So 1 sample per single smart cell. So again, this is at cost. So for folks that would have the actual machine themselves, it costs more obviously at service providers. So at people that are providing the services. But on the Sequel IIe platform, it's somewhere around $1,300 USD for a single sample on a single smart cell. And you saw the results from that from Dan. And then on the Revio platform, it's actually lower than that because the consumables are lower, and it's at $995 USD for a single sample on a single Revio smart cell. But you get 3 times the amount of data. So we're at one more question here in the last minute. Okay, so this is a good question here at the end. Is it possible to complement HiFi sequencing with short read technologies? You can. However, there have been a few publications that have come out literally in the past year as well that have done this, and they have shown that it provides no additional benefit. Since HiFi has so much higher accuracy now, the added benefit of the short reads is not necessary because there's no need to increase accuracy. And in some cases, they actually found that it actually made the assemblies worse by combining the short reads with the long reads. All right, so with that, I would like to close this out. So I'd like to thank Annabelle and Dan for their time today and for sharing their expertise and experience. Just as a reminder, immediately following this webinar, you'll receive a brief questionnaire, so please do take the time to fill it out. For those that do fill out the survey, we will select a winner for a free PacBio HiFi sequencing project to be run at Zymo Research. So thanks again for joining us today. I hope you'll join us again on a future webinar, and with that, take care and have a great day.